Welcome to another episode of the Nuclear Medicine, Molecular Medicine podcast, and we're coming to you straight from Vancouver, the Society of Nuclear Medicine meeting, and we've got some gentlemen here from Ratio, and they've been doing some great work on theranostics, something that we're very keen on in the podcast, and in particular they've been doing some great work with alphas, which again, something we've talked about before in the podcast, and I think a really exciting area that seems to be growing rapidly. So I might hand it over to you guys, if you can introduce yourself a little bit and tell us a little bit about where you're from and what your background is.
Sure. Jack Hoppin, I'm a mathematician by training. I've worked in nuclear medicine for a long time, and worked on developing an instrument, a microspec CT, and then got into really data processing and core competency. Started a company in 2008 called In Vitro. Grew that company. It's kind of a staple now in translation.
In Vitro would be probably the biggest company for really data repository and CRO development. I think in terms of imaging groups, I'd say it would be the biggest, wouldn't it?
Yeah, there's about 400 people there. It's a big organization. I left there about a year and a half ago. We sold the company in 2017 to Konica Minolta, and I started working. I'd met the good Dr. Babich a long time ago, and we connected in 2018 and got very excited about what he was doing at Cornell in terms of really thinking about radiopharmaceuticals, in my opinion, the right way, which is optimizing the kinetics of the drugs, and so helped his company Noria. And was active with him. And after I moved on from Konica Minolta Precision Medicine, started working a lot together. And I'll let John pick up kind of how we created the Ratio.
Yeah, so
from there.
John Babich. I am the Chief Scientific Officer at Ratio. My background's in pharmaceutical sciences for also 25 plus years. Reacquainted myself with Jack because one of the things we're trying to accomplish is fine-tuning molecules to really control the PK. Right? So at the end of the day, targeted radiotherapy is very similar to external beam radiotherapy. You have to make sure you hit the target, minimize off-target radiation. And we had developed a technology that's related to the manipulation of PK based on albumin interactions. And one of the things I thought it would be really valuable is to actually try to model this phenomenon, because with enough data points we could think about how we might optimize the design, not necessarily that math would drive the molecules, but it would drive the characteristics of the molecules.
Right, so what you're trying to do is optimise the uptake, not just based upon whether you get more target to off-target, but also around the half-life of the tracer.
Right, and also quantitatively get more into the tumor, right? So when you think about it, we inject a bolus of radioactivity. It would be brilliant if it were to all go to the tumor, but it doesn't, right? It all circulates around, some of it drops itself here and there, excretion, etc. But how can you control the phenomena? Two things. One is, how do you get more of what you injected into the tumor? And then how do you discriminate tumor from the rest of the tissues in the body? So it really has driven that. And Jack's analytical background was like, I wanted to find the group that could actually help us put together the mathematical basis for what we were seeing in the biology, and then that would feed back to how we would make the next molecules and so on and so forth. So that was a Noria phenomena that's continued now into Ratio. We eventually sold Noria to Bayer Pharmaceuticals back in June of 2021.
Right, because you've got to manage that mathematics, because they've all got different. You've got different uptake times from not just from the blood, but the blood into the
the half-life of the isotope and so on. So it lends itself to great. So we like to say that what we're trying to do is bring math to medicine, right? So math meets medicine here, and Jack's background and the expertise that we're pulling together between the people that Jack has brought into Ratio, the people I brought into Ratio, we're really putting together, I think, a top team to allow ourselves to really focus on the let's call it the physiological requirements to do optimal targeting. We have a lot of med chem, we've got great chelate chemistry, and it's a team that's evolving I think, to be the best in class for radiopharmaceutical development.
Right, because you've also got to look at what is out of the tumour as well.
Exactly.
What's in it, and you've got to look at what's between the tumour and the bloodstream. So you've got to look at the tissue that's sitting in the middle of the tumour that might be more insulated, more difficult, all of those kind of things. And all of those things takes math, don't they?
Yes, exactly. And even the dosimetry, particularly when you get to alphas, right, there's one thing to look at a tissue level, organ level, but there's another thing to look at a microscopic level. And that's another thing that we have to model as well. What's happening at the very level of the tumor cells and its stroma.
Well, that's really important because that's one of the key advantages of theranostics. You can't give a radiotherapy beam at a single cell. You can't even do surgery on a single cell, really. The only way you can actually treat a single cell of cancer, and that single cell might become a few trillion cells in a fairly short period of time, is with a theranostic approach, or a chemotherapy approach, but again, a chemotherapy is indiscriminate in terms of it.
Exactly. No one typically knows where chemotherapeutics go.
Exactly.
It's not a question that's asked too often.
Yeah, I think what we're touching on here is a key that is true to this space. Is it's a very interdisciplinary space. You know, you really do need to build a team. Having both of us been in the field of nuclear medicine for a long time, but also been worked with a lot of non-nuclear medicine pharmaceutical companies. Very often, everyone thinks, well, if I just put an isotope on something, I have the drug. You know, I've had this drug do something. All I got to do is put an alpha or beta emitter, and then I have a drug. Or in nuclear medicine, sometimes there's a sentiment where I just need to find a binder. I already know how to make radioactive drugs. But the truth is, you really have to have kind of a comprehensive understanding of drugs, nuclear medicine, and really physics, and in this case as well. And I think that's part of our goal. We have
timing,
and the market. Yeah. We're incredibly appreciative of. Right now is a great time. We're seeing the adoption because of the success of some of the new therapeutics. You know, and large-scale pharma moving into this space, and that's not something we saw 8 years ago. You know, now is a new era for targeted radiotherapy. It's just a new time. It's exciting,
and it's happening not because the big pharma want this to happen. It's because they've been dragged kicking and screaming, I think.
Yeah, and well, I mean the patient benefit is so good and equivocal.
Yeah.
Well, you know, we've spoken before with people like Josh Malman, who's a patient advocate, right? And so they're the people that are driving the need for this and then saying, look, this works. We want what works.
Yeah, and the side effects are typically much less. And it was everyone's kicking and screaming, not just the pharmaceutical companies. I mean, clinicians also. No one really wants to work with radioactivity, but I think we're making it very. You know, it's so valuable that it's getting better and better and more adopted. And supply chain distribution, it's all going to. You're watching it all. The ecosystem, exactly. So and a lot of investment has moved into the space, and so the infrastructure will also rise.
Okay, let's talk with specifics about what products you've got in development or about to get into development. I was particularly interested in the FAPI. Can you tell us a bit more about that?
Sure. So, you know, FAPI is a great target, right? It's a unique target in that it's really not a cancer target. It's a tumor target. Right. You might say, what's the difference? Well, the cancer cells are the actual cancer itself. The tumor is the swelling, right? The Latin for swelling of what is the mass of the tumor. And so I like to think of it in sort of simple terms. It's kind of the bird's nest, and that makes up the tumor, and the eggs are the cancer cells. And so the nest is an important part of the environment that allow the tumors to grow. And so sometimes there's more nest than tumor cells, right? So we're seeing now the development of the theranostic piece on the diagnostic side, saying, wow, this might be a better way to find tumors, characterize tumors, because there's a lot of signal there. So you can there's a lot of data talking about comparing it to FDG, but there's other things to it as well, and that it's really looking at a component within the stroma, which is the activated fibroblasts, which do a lot of things, right? They're controlling a symphony of things in the tumor, and some of that may be related to immunosuppression or the covering up of the tumor from the host immune system. So that it opens itself up to being a very interesting target therapeutic.
It's not
lots
just,
of tumors have it.
Yeah.
And so it's, it may be pan carcinoma at some level. But it's also going to be interesting, and I think we're at a very, very early stage here. What is the impact of destroying the stroma? Is it a way of breaking down barriers, physical barriers, or physiologically to get more drugs into those tumors? Say pancreatic cancer, which has a huge amount of stroma component. It's very hard to get drugs into pancreatic cancer to get the cancer cells. Is it also a way of shutting down or pulling away the veil of the immunosuppression in order to activate
the immune therapy.
Exactly.
So you're revealing the DNA. You're breaking the actual
you're going
tumor apart.
To get more access, right? It's an access issue, and so if you look at the literature, there's not a lot of discussion about drug delivery. There's some very bright guys who, in both times, listen. We still don't know where the drugs are going, and so one of the issues may well be drug access. This could be an adjuvant for lots of different cancers by basically pre-destroying the stroma before you get into a therapeutic basis. Right. So there's lots of opportunity here, and the game's got to get played.
And a lot of theranostics fail, or treatments fail, or even FTG fails because they're just looking at metabolic activity of the tumour, right? Whereas the stroma is you don't have to have a high metabolic activity to have a significant amount of stroma around the tumour.
Exactly right, which is why very often, the data and the theme of this conference right now is a lot of presentations around this was a silent tumor in FG, and now I'm seeing a FAP signal.
Yep.
And
it was Image of the Year a few years ago. We did a podcast exactly on the Image of the Year.
Yeah, there you go.
So
exactly. But now that was the hope, and we're starting to see the promise realize this. We're starting to get more and more data.
And it hasn't taken that time, though.
No.
Dr. Geisler did Image of the Year a few years ago with FAPI, and that wasn't that long ago.
No.
And we've now got a theranostic. Now, if that's going to happen with this, what's going to happen with HER2, right? Breast cancer, right? What's going to happen with some of these other cancers? Are we really going to? We're going to significantly make. Often the cancers that we've come out with theranostics, like neuroendocrine tumours and slow-growing prostate tumours, are the difficult ones.
Yes.
And we're having success in the difficult ones. So I think this is great news for patients.
Yeah, so we're excited, we're patient-focused, and it's all about the patient at the end of the day. Can we extend life with quality?
Yes.
And that's a huge opportunity for theranostics. It's just, the lack of side effects from most of the therapeutics, they're not going to be zero, but they're going to be really quite I'd say gentle relative to chemotherapy and other things.
So the alpha emitter's got an incredibly short pathway. So it barely gets across the cell, right? So it's really not going to affect neighbouring cells very much.
You just have to get it in the right place.
Exactly.
You just have to get it in the right place.
Yeah. I'll add that we're, just from a kind of where we are as a company, we've been. It's kind of our first announcement
display.
We started the company a year ago. We're 15 people, heavy PhD at this point, kind of 13. We really have a senior scientific leadership group as the company grows, and we move into a headquarters lab, discovery lab, in January of next year, 19,000 square feet in the seaport of Boston. So it'll be, radiochemistry, synthetic chemistry, rodent, preclinical discovery work, and our headquarters office-wise. So we're very excited. We got a good foundation, and looking to do this for a long time. It's not and hopefully, again, we have good partners already in Bayer and Lantheus, and I think we're very excited.
It takes the ecosystem.
It does.
About, right?
Yeah.
You have manufacturing. I still manufacture capabilities with Lantheus. We've got a huge pharmaceutical powerhouse in Bayer, and it's we're very fortunate that we have those two relationships and great partnerships.
Right. And the other thing is we're now starting to come online availability of the source material, the alpha emitters. Like
exactly.
There's a new development at ANSTO in Australia.
Exactly.
Right.
Yeah. And we just talked with one of the ANSTO colleagues yesterday at we were at Triumph.
That's right.
BDOXT, and talked to BDOXT, what they're doing, and people from ANSTO. So there's a huge amount of energy we're putting in financial support, obviously, to make this happen. It's a really exciting time.
Excellent. Yeah, it's been great talking to you.
Likewise.
Yeah. Anything else you'd like to add? Where can they find out more about what you're doing?
Just our website, and we just kind of launched, so that was our first press release. We'll kind of keep the message going, and we
info@ratio.com.
info@ratio.com, ratiotx.com.
All right.
And we're here and looking for collaborators too. So
Excellent.